All right, so today we're going to start our section on DNA, and with our section of DNA, what we're going to be looking at are two different note sheets. So the first one you see here is just the notes itself, and then you guys are also going to see one that goes through on diagrams, and you'll see them underneath the listing on Google Classroom as well. So I'm going to kind of be flip-flopping back and forth, so as I go through it in words, I can try to show you what it looks like within pictures there. So that's what we're going to be looking at today. I'm going to switch my camera around a little bit for you guys. And let's start with some of our written stuff. So, as we start talking about DNA, the key thing here to remember is that DNA really wasn't discovered, Nobel Prize. We're talking like 1950s, 1960s, which truthfully isn't that long ago when we think about things that have occurred within science and how quickly things have evolved since then. We're cloning organisms since then within the last, 50, 60 years. And if we think about how long it took us to get to a certain point and how quickly we're moving now, it's pretty incredible. So some of the scientists that helped us out with this along the way are ones that you guys are going to see here. So we're going to talk about them, and then I'll come back to them where it says, hey, this is page one of the diagram packet. We'll go to that page one of the diagram packet and kind of clarify some of the stuff with you guys. So the first two scientists are known as Griffith and Avery. You guys are going to see a little worksheet that goes through these guys as well at the end of this particular lesson. So that will kind of be a sign for you guys, and you'll see some more about their particular experiment. So these particular scientists, about 1928 is when they started going through and actually looking at some of the stuff that we have here. Now people were really going, hey, let's go through and study stuff on DNA because they weren't really sure what was being passed along, what is giving us our traits, what is the genetic material. So they worked on what was called the transforming factor. So what they worked with were mice, and they worked with this Streptococcus pneumonia bacteria. And what happened here is they worked with S strain bacteria and R strain bacteria, which is smooth and rough coat bacteria. So I'm going to kind of flip there, and so we can kind of look at see what we have. So when we're looking at this particular one, this transforming factor, meaning this changing factor that we're looking at, these two scientists point, they weren't looking at DNA. They were actually working on a vaccine for pneumonia because at that point, pneumonia was killing a lot of people, 1920s at this particular time. So what they did is they found a couple different strains of the pneumonia bacteria. One was called S, one was called R. It's really light there, but you'll see that that's the R one. You guys can also follow your letters up above. The S and R are your smooth and rough coat bacteria. So what happened is they took live S Streptococcus bacteria, the smooth coat bacteria, and they put it into the mouse. And they said, hey, this type of bacteria is virulent. It's deadly because you can see the little mouse is dead. The live R, the rough coat Streptococcus pneumonia bacteria, actually is non-virulent. So when they put that into the mouse lived. So they go, okay, well, let's do this. If we take that virulent, that deadly S-type bacteria, and we heat it to a high temperature, because we know heat will kill that bacteria. If we heat that deadly bacteria to a high temperature, we say that we attenuate it, and then we put it into the mouse. What they saw is the mouse lived. So live S kills, the heat, high temperature heat killed S strain does not kill. So they go, okay, well, how about if we take that live R, which was non-virulent, non-deadly, and we take this attenuated heat-killed S strain, which didn't kill either, and we kind of put them together. And when they did that, even though the live R was non-virulent and the attenuated strain was non-deadly or non-virulent at that particular time, when they put the two of them together, what they saw is the mouse died. But what happened, type R, this rough coat bacteria, was transformed. So even though this bacteria is dead, the genetic material, they didn't know this at the time, the genetic material inside of it kind of protected itself. So where it wasn't able to kill in this particular form, here it took over this non-virulent bacteria, took over the nucleus, and transformed it into that deadly type. So that's where it was actually able to kill that particular mouse. So this transforming factor means that the DNA kind of changed in another cell there. And that's what they were showing with this particular experiment. So those are your first scientists. So that would be Griffith and Avery, once again, about 1928. And I'm not going to test you guys on dates of any of this. I'm just trying to give you guys a little timeline to see, hey, this is where it started. weren't really talking about DNA. Maybe this is where we were starting to talk about DNA as we move on. So the next one, we have Beadle and Tatum. So you guys may have heard this term before where we talk about one gene, one enzyme theory. And that's something we mention a lot of in first year bio, one gene, one enzyme theory. So these scientists identified genes as specific units that function in heredity. So a segment of your DNA, which codes for a protein or a trait. So that segment of your DNA is a gene. That gene, that segment of DNA, is going to code for one enzyme. And we also know enzymes, proteins. So sometimes you might hear this little theory as the one gene, one protein theory, or the one gene, one enzyme theory. It is the same thing. So when we're looking at these particular guys, and we'll look at our packet here in a moment, they were actually working with red bread mold. And they were looking at these different amino acids that they were either to pull out or put in to see whether or not these particular things were able to grow, this mold was able to grow there. So if we're looking at our particular diagram, we're going to go to page 2 here. And there were a couple scientists that kind of went through and did this. So they actually Beadle and Tatum actually won the Nobel Prize for this in 1958. This whole idea of one gene codes for one specific enzyme in your body. So they take this red bread mold and they start putting different things into it and taking away different things. And then what they saw is that mutants cannot grow without supplemental amino acids. Well, we know amino acids make up our proteins, and proteins are, or enzymes are special types of proteins. So if they're lacking something, we can't put these things together. We can't make this string of amino acids to make that particular enzyme. If you lack the gene to make the enzymes, then you can't allow that particular growth. So if you don't have that segment in your DNA, is another thing they were looking at. If you don't have that segment in your DNA that says, hey, let's put these, this is how the code goes together in order to make that, then we're not able to go through and do it. So they'll lack the gene to make the enzymes to allow that particular growth. But they were also grown without particular amino acids. So if we continue a little bit, here's some of the things that they have. Here were different mutants that they created, and these are different amino acids that they were putting in. So we were looking at arginine was one of the big ones that they were doing here. So we have our medium, we have some other little things that they were putting in, supplemental organic salts, maybe some carbohydrates that allow them to grow, and they were putting all of these in here. And as they keep going through, they had to make sure that, hey, we need certain materials put together in a certain fashion in order for that protein to be created. So one gene, one polypeptide, same thing as the one gene, one protein, because we know a polypeptide is also a protein, which is also an enzyme. So an enzyme is just a special type of protein. So other scientists kind of went along with this and started looking at some of these ideas as well. So Luria and Delbruck, you guys may have heard of something called the Hershey-Chase blender experiment. So these two scientists, and they always kind of worked in pairs here within the labs, they actually studied viruses and bacteriophages. Well, hopefully you guys realize that a bacteriophage is also a virus. It's a virus that affects bacteria, and scientists love to work with them. They're usually larger than normal viruses, and they only infect bacteria, so you don't have to worry about them infecting you. Bacteria is super easy to grow. It doesn't cost a lot to grow within the lab, and they were trying to see what part does the virus actually transfer on. Does he transfer on this outside portion, which is the protein coat, or does he transfer the material that's on the inside of them? What was the genetic material? And that's what they didn't even realize. Back now, it seems like, hey, that's pretty easy. It's the genetic material on the inside that gets passed along and gives everything its traits. But way back in the early 1900s, they weren't really sure. This is about 1950s now is where we're with this. So they actually will tag, meaning they mark different parts of this bacteriophage. So the bacteriophage is kind of like that little spider guy. He's got the little capsule head. He's got the little tail fiber legs. And they tag different parts of them so that they can see what was getting transferred along when it infected that particular bacteria. So they tagged the DNA with one radioactive type, and they tagged the protein coat with another radioactive type. And that way they could trace what was being passed on to that next generation when they infected it. So if we look at that particular diagram, so once again, here's my little bacteriophages. You guys see them right here. And these viruses were tagged with radioactive isotopes because those isotopes are what they could go through and monitor. So the protein coat, the outside portion of it, the capsid portion of it, they actually marked with radioactive sulfur. The genetic material, which we now know as genetic material, which they didn't know then, but the stuff on the inside of this virus, they labeled with radioactive phosphorus. And then they kind of put them into solution. They allowed those viruses to infect bacteria generation. Well, what's going to happen is that whatever material that they pass through to that next generation is also going to be copied, which means that my radioactive substance, whatever it may be, is going to get copied and go through that. And so what we see here is that we have phosphorus being passed through, and at the end, there is no genetic makeup or no genetic information of what we see here with our radioactive sulfur. So it wasn't the protein code that got passed through, but it was the genetic material on the inside, which is your DNA. So my radioactive phosphorus is what was getting picked up. So what they were looking at here is, with this blender experiment, is that the genetic material, the inside stuff, is what had the information that got passed on to that next generation there. So a couple scientists kind of went along with that and looked at it. So we talk about this, Luria and Delbruck. We talk about Hershey and Chase, but it's the blender experiment. So it's not like, hey, these two scientists, they're just doing this experiment. There are scientists all over the world at this particular point. They're kind of all trying to figure out the same information. They're on the edge of something that's so new that so many people were trying to study at that particular point. One of my favorites is Rosalind Franklin. And the reason she's one of my favorites is because she's one of the first females within science that we really talk about within biology. And she's the first person to actually use something called X-ray crystallography. So she actually takes a photo of the double alpha helix of DNA. And this is actually her famous photo here. So she's kind of looking at it from the top, taking this X-ray crystallography of it, and she could say that it is kind of this double alpha helix because at this point we're going, okay, well, we have this genetic material. We kind of know what it's made up of a little bit, but we really don't know what it looks like. And once we figure out the structure of what it looks like, then we can really start saying, okay, how does it come together? How does it get transferred along? Does it replicate? How does it do this? So they need to kind of fill these pieces in. And Rosalind Franklin is one of those particular individuals that does this. So she kind of worked kind of closely with Watson and Crick, but was not a friend of Watson and Crick's, unlike what they thought. So she wasn't sure how the bases or the strands really worked within her particular model. The problem with Rosalind Franklin, so she's working with X-rays, and we're talking in the 30s and the 40s. X-rays back then, super dangerous. X-rays right now, super dangerous. But we have learned how to kind of cone in X-ray views. We know to wear those leg gowns so that it doesn't affect our cells. Because what happens is that X-ray can lead to damaging of our DNA of our cells. It can lead to cancer, which is exactly what happened to Rosalind Franklin. She actually passed away of cancer when she was in her, I want to say like mid 30s. So before she was actually able to go through and really kind of enhance her information. But we're also going to talk about how Watson and Crick kind of stole her photograph, took her information, and came up with this whole idea of, hey, DNA is this double helix, and never gave Rosalind Franklin any credit for it. So where Watson and Crick go along and they win the Nobel Prize for it, Rosalind Franklin never gets any credit for this, even though she's the one that came up with this picture of it that led them to this idea of it. All right, so let's go through Levine. Before we get to Watson and Crick, let's talk about the parts that make up this double helix. And we've talked about nucleotides, and we've talked about purines and pyrimidines, so this stuff should kind of now, hey, let's pull it from the back of the head and pull it forward now and use it a little bit more. We did it with organic when we talked about nucleic acids and some other parts. So a scientist by the name of Levine, he actually studies the structure of a nucleotide base. So nucleotide, one sugar, one phosphate, one nitrogen base. And the nitrogen bases that we're going to look here are called purines and pyrimidines. Purines, if you guys remember, and you'll see them within the diagram packet, are made up of double ringed structures. So those are those larger base pairs that make up the rungs of the ladder. So if these are the sides of the ladder right here, these are the rungs that we walk up on. I need two base pairs to make up those rungs. One of them is called a purine. It's larger. Let's go with my smaller finger. One of them is called a pyrimidine. It's going to be a smaller base pair. So my purines, my double ring structures, are adenine and guanine. And this helps us to kind of pair things together. I always want to pair one purine with one pyrimidine. My pyrimidines are single ring structures. They're kind of smaller, so that would be like my pinky versus my index finger. So my purine, my pyrimidine, my adenine or guanine, my thymine or cytosine. And we always, like I said, put one purine and one pyrimidine. Before we go on there, though, how do you know which ones to go together? So why can't, if one purine, one pyrimidine, why can't I put adenine with cytosine? There's one purine, there's one pyrimidine. We know adenine and cytosine don't go together. It's always A and T and C and G go together. And the reason we know that is because of what is known as Chargaff's rules. So Chargaff actually went through and kind of came up with this idea like, hey, every time I see adenine, I see the equal amount of thymine within the structure. That must mean they pair together. Well, how are they pairing together? Oh, they have two hydrogen bonds between them. So the only way that I can pair up these nitrogen base pairs is in this particular fashion. Adenine, my purine, has to go with thymine, my pyrimidine, because they have two hydrogen bonding spots together. Just same thing with cytosine and guanine. Every time he looked at this structure, he said, hey, I have equal amounts of cytosine as I do guanine. They have to be in equal amounts. Why? Because they pair together. And they have three hydrogen bonds together. So I always will have equal amounts of adenine and thymine, one purine, one pyrimidine, and equal amounts of cytosine and guanine. So I could say, okay, well, within a DNA strand, maybe that strand, 10% of it is adenine. You should be able to calculate the rest of it. So if 10% of that strand is adenine, I know 10% of that strand is thymine because they have to be in equal amounts. They pair up together. So 10% of adenine, 10% of thymine, that gives me 20%. That means I have 80% left. Well, that 80% has to be split between cytosine and guanine. So that means cytosine has to make up 40% of that strand, and guanine has to make up 40% of that strand, and they equal up to be 100. So you need to know how many bonds are between adenine and thymine. Two hydrogen bonds are super weak, and I have three hydrogen bonds between cytosine and guanine. Going on to Watson and Crick. So Watson and Crick, so when we think about DNA, it's like, hey, Watson and Crick, these are the great guys. They've come through, they've figured out DNA, what the structure looks like, they win the Nobel Prize. Yeah, they actually stole it from Rosalind Franklin, and they weren't exactly the nicest people when they went through and did that. Like I said, they gave them no credit for any of the information that they came up with. So they actually these two particular individuals come up with the structure of the double helix in 1953. They win the Nobel Prize shortly after that. And later on, Crick keeps going through, and he actually comes up with the theory of the genetic code about 10 years later, saying that, all organisms have different genetic codes, and our codes give us all of this, and this is what they do, and so on. So if we look at our next little diagram here, so this is where it kind of goes, ooh, now we got a lot of stuff going on. So we're going to break this down, and we're going to see it over and over and over again. So I do like these diagrams. I'm going to start at the top, and then I'll kind of work my way down because there's a lot of stuff at the bottom. There's a lot of stuff that you guys are going to see as we get into our DNA structure notes as well. But it's still dealing with scientists here. So Watson and Crick, they come up with this shape of DNA. It is a double helix. It is a ladder that's twisted around. Like I said, they won the Nobel Prize, but they were helped by Rosalind Franklin. She doesn't get any credit even after she passes away. And she did the whole X-ray crystallography, which took the picture from above, showing that it is actually two strands that come together. Because at that point, they weren't quite sure how all these particular parts came together and how it got transferred on. All right, so let me grab my little diagram here. Structure of DNA, and I've got one here in front of me as well. So my DNA structure, and you guys probably built them in first year bio, kind of looks like this. So there is my structure. The tan pieces along the side are the sides of my ladder, and then in the middle you see the blue, the yellow, the green, the red. They are my nitrogen base pairs. So what's going to happen here is that they kind of spin around to make my ladder. I'm kind of switching it so that you guys can see it's just a straight ladder there. There is my double helix. The outside of my ladder is made up of sugars and phosphates, and they alternate sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down along both sides. The rungs of my ladder, kind of like if you were walking up a ladder, this is where your feet would go to, are made up of my nitrogen bases. So if I said, hey, let's look at a nucleotide. A nucleotide is one sugar, which is this orange piece here. In DNA, it's deoxyribose. One sugar, one phosphate, one nitrogen base. So this entire piece right here would be my nucleotide. In DNA, I have 4 different nucleotides: sugar, phosphate, guanine, sugar, phosphate, adenine, sugar, phosphate, thymine, and sugar, phosphate, cytosine. So since I have 4 different nitrogen bases, I have 4 different nucleotides, and these nucleotides come together to make the structure that you guys see in front of you now. So off to the side here, you see the nucleotide: sugar, phosphate, nitrogen base. In DNA, my sugar is deoxyribose, so you can actually see the little D's here. So P, D, P, D, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. What you're going to need to know is that your nitrogen bases will always attach to your sugar. So you can see here cytosine is attached to my sugar. So sugar, phosphate, sugar, there's guanine, phosphate, sugar, there's my next base pair. You can also see in the middle between them, I have my hydrogen bonds that are extremely weak. Chargaff's rules, we just talked about them. Chargaff said that equal amounts within that particular strand of DNA. Every time I see an adenine, I have equal amounts of thymine. There's 2 hydrogen bonds between them. So A and T will equal each other. They always pair up unless there's a mutation. And cytosine and guanine will also always pair up. They have 3 hydrogen bonds. And that's how you know which base pairs are complementary to each other, which ones match up together. Levine's the one to talk about these nitrogen bases, your purines and your pyrimidines. So I'm going to scoot down a little bit. So we change our view that you guys see here, this nice little ladder, and all of a sudden it gets really complicated down here. What we're looking at is the chemical makeup of it. So we just took my sugars and phosphates and nitrogen bases and said, hey, let's break them up. So here is still the backbone of my ladder. There's my phosphate, there's my sugar, there's a phosphate, there's a sugar. How do I know this is a sugar? Look at it closely. Well, I have carbon, hydrogen, and oxygens, and that's it. We know that that's what sugars have in them, those 3 elements. I know I'm in a ring form. There's my ring. And we're going to talk more about that ring as we talk about 5' ends and 3' ends a little bit more tomorrow. This is my nitrogen base. I should be able to start to look at these and know which nitrogen bases they are. You go, well, how the heck am I going to do that? First off, I know they're nitrogen bases because they have N in them. They have that nitrogen in them. Here is my smaller base pair. Here is my larger base pair. So we talked about purines and pyrimidines. We said purines are larger base pairs. They are double base pairs, double rings, 1, 2. So there is my purine there. It always pairs together with a pyrimidine. So 1 purine, 1 pyrimidine are making up the rungs of the ladder. So I know that this is my pyrimidine. So how do I know if it's guanine and cytosine, or I know that it's adenine and thymine? Well, now I go to my hydrogen bonds, which is what Chargaff is looking at there. Here I have 3 hydrogen bonds, and this is where you have to memorize this stuff. This is basic memorization, unfortunately, but it's going to allow you to apply that information as we build upon this particular structure. So I know that cytosine and guanine have 3 hydrogen bonds between them. So this has to be guanine because it's purine. This has to be cytosine because it's pyrimidine, and they got 3 hydrogen bonds. Look at the next one, 2 hydrogen bonds. Double ring structure has to be adenine. Single ring structure has to be thymine, A and T, 2 bonds between them. Here I have 3 hydrogen bonds, a smaller base pair, a larger base pair. Here's cytosine, here's guanine. You're also going to notice that your nitrogen bases are always attached to your sugars. Always, always, always will be attached to those particular sugars. So the thing about DNA is that it is a huge molecule. So when you go, oh, DNA is really small, you find it in your nucleus. It's actually a very giant molecule in terms of molecular. It's found in the nucleus of all cells of eukaryotic organisms. Bacteria still has DNA, but it has it kind of in that little nucleoid region where it's not really protected by that nucleus. And that nucleus within these eukaryotic cells kind of keeps it trapped within. Remember, this structure, this DNA, codes for all your proteins. Okay, it has a gene, each segment of it. So this piece here to this piece here is going to code those A's and T's and C's and G's, code for maybe a particular protein or an enzyme for you or a trait for you. That's going to happen out into the cytoplasm. However, this guy can't get out of the nucleus. How are we going to get him out into the cytoplasm? And that's why we have to go through this whole process of protein synthesis as we go through. Before then, we're going to talk about, okay, well, I need to make more cells. I'm growing, I'm repairing, I'm developing, I need more cells. Well, in order to do that, I need to replicate this DNA. I need to make an identical copy of it so that it can go to a different cell. And that's where we're going to talk about this idea of DNA replication. But once again, DNA is found in the nucleus. It's too big to leave through the nuclear pores. It is a double helix. So we go through this whole idea of DNA replication. We're going to talk about how this nuclear envelope kind of breaks down for a short period of time so that my DNA can replicate. It contains twice the amount of DNA. That cell is able to divide, and then the nucleus kind of reforms around them. So we'll always kind of be within that nucleus unless the cell is actively dividing. You guys see once again with Levine, here's my purines, double ring structures. So this is what they look like, but chemically, here's my first one, guanine. Here's my second one, adenine. My pyrimidine, single ring structures. Here's thymine. There's cytosine. When we get to RNA, we're going to talk about another nitrogen base pair called uracil. Uracil is just going to replace thymine. It does everything thymine does. It's still a pyrimidine, a single ring structure. It's still going to bond to adenine with 2 hydrogen bonds in it. It's found in RNA. Thymine is found in DNA. All right, the next thing that we're going to talk about, I'm just going to introduce this real quick in this particular section so we can think about it, but we're really going to talk about it in the next section because it gets a little bit more complicated. And first year bio, some of you guys talk about it. I know we did it within my honors classes and Mr. Blue's honors classes. I'm not sure what they do in the next level. So DNA has direction. So when we talk about this double helix, this ladder, they have running in opposite directions when it gets made and when it gets read. So DNA, we say, has a 5' and a 3' end to it. So what is 3'? What is 5'? What does that actually mean? Well, it deals with the number of carbons within it. Remember, deoxyribose and ribose is a 5-carbon sugar. So it's kind of similar to glucose, which is a 6-carbon sugar, but it is a 5-carbon sugar here. So when we're looking at my sugars, you'll know which one is sugars because it has a ring structure to it. It makes up the backbone of my ladder, and it's always attached to a nitrogen base. It doesn't matter which sugar you look at. We're going to look at the one on this side right here. So I'm pulling out this sugar, and what I want to do is I want to highlight my oxygen. So within that ring, I'm going to find my oxygen. And then I want to count my carbons clockwise. So I'm starting here at this little red arrow where my 0 or my oxygen is highlighted. There's my first one, there's my second one, there's my third one. So my third carbon is facing down. So I know the bottom of that strand is what is known as a 3' end. If I keep going, that was my third carbon. There's my fourth carbon. My fifth carbon is facing upward. That means the top of the strand is my 5' end. So let's see if I have a little pointer here that may help you guys out. Okay, so once again, we're looking at this one here. There's my little oxygen, and that's where we want to count. So we said if we counted them, 1, 2, 3, the bottom here is going to be my 3' end. That's what we have down here. We keep counting my carbons, 4, 5. My fifth carbon is facing upward, and that's what we have. Well, if we go to the other side of my strand, which is this strand over here, so we're going to go all the way over, find another one. I don't care if it's this one you do. I don't care if it's this one you do. I don't care if it's this one you do. They're all going to be the same. I'm going to do one down here since it has red arrows on it. Same thing. We're finding the oxygen. In this case, the oxygen happens to be at the bottom of the ring here, and we're still going to count my carbons clockwise. So if I'm counting them clockwise, my first carbon, second carbon, third carbon. And that's all we're worried about is the third carbon and the fifth carbon. Here, my third carbon is facing the top of the strand. So I know the top of my strand is 3' end. If I keep going, 4, 5. The bottom of my strand is my 5' end. So my strands run in opposite directions. This one goes 3' to 5'. This one goes 5 to 3. And why is that important? Because DNA When we replicate it, those strands get read and made in a certain direction. So when we kind of split these bonds up here in a moment, DNA, the enzyme that's going to come through, DNA polymerase, is going to read the strand in this direction. So whatever base pairs start here, it's going to go up in this direction. This strand is going to get read in this direction. So both sides will get read, 3' to 5', but they're running in opposite directions. They are said to be anti-parallel with it. Okay, and we'll see them a lot more when we get into some of the notes. So we will come back to them, just kind of putting it in your head for now. But when we get into that DNA replication note, that's when we're going to see that. Some other scientists you guys are going to see along the way. I'm just going to kind of finish up the scientists within this section, because then the rest of those diagrams are really going to get into as we get into our DNA replication notes. Meselson and Stahl. Meselson and Stahl come up with this idea, and this is definitely a word you guys should have heard before. Semi-conservative DNA replication, or semi-conservative replication. So when DNA replicates it, it makes a completely new copy of itself. That new copy, or that new strand that we just created, is made up of 1/2 old and 1/2 new DNA. So in other words, if this is my original DNA strand and it has to open up in order to make new strands, new base pairs are going to come in, I now have two new strands that are completely identical. Each strand has 1/2 old DNA, 1/2 new DNA in it. Semi-conservative replication. I had to use the old strands as kind of like a template or a mold so I know which base pairs come in together. And that's what Meselson and Stahl was looking at within their particular um, diagram. And you guys will see that when we get to the DNA replication one. Sutton, you'll hear a little bit more of as we work our way into chromosomes. Same thing with Morgan. He basically describes chromosomes as this true unit of heredity. So chromosomes, which is made up of our DNA, is what's kind of getting passed along from one generation to the next. So he looks at grasshopper chromosomes during the process of mitosis and meiosis and sees how things kind of get passed through. And then another scientist, Stevens and Wilson, first identified male and female sex chromosomes. So as they're going through, this is that whole idea of karyotyping comes in too. And what they notice is that that last little set of chromosomes were a little different between males and females. Morgan, we'll hear a ton of when we get the genetics, because we'll look at Drosophila melanogaster, which are your fruit flies. And Morgan actually started a fruit fly room at Columbia University way back in the early 1900s. And here's the deal, it still exists today, and they still have the kind of same genetic makeup of what they started with, meaning those first flies that they started with, they're still breeding that generations of flies there. And what he looked at is flies traits as well as mutations. And some of the mutations that you guys are going to see, we'll look at ones that instead of having their little antenna, they have antennapedia. Looks like they have little feet on their antenna. Their antennas are really big on the top. They'll have curly wings. Apterus, where they have very little wings and they're not able to fly. We'll look at different eye color instead of like a normal brick red color. Maybe they have yellow. Maybe they have white eyes. Different eye shape, whether they're bar shaped or whether they're nice and round. So we'll look at a lot of those different fruit fly mutations when we get the flies. Hopefully we'll be back in the room a little bit so you guys can look at them under the microscope and kind of put them in a little fly nap gas, a little sleepy ass, and put them in a little petri dish. It does not kill them because you kind of will see them moving around, and that way we'll give you guys a chance to be able to identify a lot of the traits that we see. Those who are virtual, I'll do the best that I can to kind of tape through the microscope that we have, and you guys will kind of be doing the same thing there. So that's coming up shortly, I guess, after DNA and RNA. Okay, so I'm going to stop there because that's then going to take us into the notes on DNA structure, and we'll kind of continue with that a little bit more next class, and we'll really start jumping into a lot of the ideas. The good thing is DNA structure, we just went through a lot of it. We talked about Watson and Crick, we talked about Levine, we talked about Chargaff, we talked about Meselson and Stahl, anti-parallel, running in opposite directions, 3' and 5' ends. So if you're like, ooh, that's a lot of information, it absolutely is. Here's the deal. Write down questions as you guys do this. Islam, you're going way too fast on this page. I didn't really understand what was going on with this. Can we go back and review this? Unless you guys ask questions, I'm assuming that, oh yeah, this is old stuff. We've done this before. We're just kind of enhancing upon it before we work our way into some of the other material. Okay, so please, please, please write down stuff. If we need to contact me. We'll Google Meet, just two of us, three of us. If you guys want a small little group, a little study group together, that's fine. We can go through the information. Please just let me know. Okay, so we're going to stop there. You guys are going to see one worksheet that deals with some of the scientists. Actually, we'll talk about the mice and the smooth coat and the rough coat and the virulent and non-virulent bacteria that they worked with. So you guys will see that listed there, and we'll continue with DNA at a later time. Thanks.